All right, so once again, thank you. Once again, Caleb Gimar, UTSA/Southwest Research Institute. And I'm going to be talking about my work on the lunar soils, Apollo 10084, 68501, and 71061, in terms of their far-ultraviolet bidirectional reflectance. So the key points to take away here are that we have done these measurements on these three lunar soils. We have found that space weathering effects probably have a very pronounced effect on the scattering anisotropy of these soils in the far UV, meaning that the space weathering has an effect on the direction that the soil scatters light. We do not find in this study a strong correlation with any overall compositional effects, which is a little different from what we found in comparing simulant reflectances to Apollo 10084 in our previous study. And then we want to expand this work onto hydrated lunar soil samples here very soon. So the samples in question, you see them in these photographs here. These are the actual sample trays that we use in the sample, Apollo 10084, 68501, 71061. You can definitely tell that the 10084 is a Mare soil sample, whereas the 68501 is very much a highland soil sample. The color difference is very well seen in those photographs. But of course, the main difference that we're interested in is the overall compositional difference, which you can see in this plot here. You'll notice that the main difference is in titanium-bearing mineral content. The 68501, like many highland soils, is a very low TI soil, whereas the two Mare type soils, we have 10084 and 71061, are the higher titanium soils. The other difference we are interested in here is maturity. So each of these soils give us an opportunity to probe the effects of maturity. 10084 and 68501 are very high maturity soils, ISO/FeO 78 and 85 respectively, whereas the 71061 is a low maturity soil. So we can compare high maturity to low maturity for a similar chemistry soil and chemistry for two identical, or at least very similar maturity soils. Now we have to ask, why is this important? Well, we want to build up I guess you could say an atlas of far UV reflectances to be able to utilize LRO lamp observations of the lunar surface to be able to interpret the compositional and weathering effects that may be visible in those observations correctly using backing laboratory data. So our chamber, the Slater Crater Chamber as we call it, formerly it was called Southwest Ultraviolet Reflecting Chamber, is designed to emulate the LRO lamp observation scheme in the laboratory environment. So as LRO lamp traverses over the lunar surface, the far UV radiation strikes the lunar surface and LRO lamp observes the reflected UV light. In our chamber, we have a channeltron detector mounted to a rotating armature, which observes the far UV light striking our sample in the chamber similarly. The special thing about this setup is that it allows for absolute far UV bidirectional reflectance measurements, meaning we can retract this sample tray, put the detector right in the way of the light beam, and get a measurement of the intensity of the UV light hitting the sample at the time of the measurement without having to rely on certain standardized read it from the text kind of measurements, basically. This is a far UV and I should say ultra-high vacuum chamber, so meaning that this chamber gets down into the mid-10⁻8 torr region. So this is a photograph of the chamber itself, and this is the interior of the chamber. You can see the detector. You cannot quite see the channeltron detector itself. It's mounted behind that little plastic block there on the rotating armature. Here is the retractable sample tray right here. And there are a few other features, this doser head and the QCM there, which will eventually be used for the hydrated soil measurements. And so here are results. So what you see on the far left-hand side of the slide here is the actual raw results from the chamber, and since that they've also been corrected, of course, for the collecting area of the detector and the angle at which the sample is being observed. And so you'll notice that immediately the black points, which are for 10084, and the blue points, which are for 68501, there's not a huge amount of difference between those points. They sit almost right on top of each other. And that, of course, indicates that there's not a huge difference in spectral response of those two soils. However, for the 71061, you'll notice that the phase curves of these tend to go upward some toward the far end, generally. And that indicates that that is a very forward scattering soil. And overall, these phase curves are a little higher than those for the other two soils. So what does this mean? Well, when we go through and we actually do the Hapke photometric modeling on these soils, we can actually see that overall the single scattering albedo of the two high maturity soils, 10084, 68501, are very close to each other. Again, though there is a chemical difference between these two, it does not seem to be having an effect on that overall single scattering albedo. The 71061, however, the lower maturity soil appears overall quite a bit brighter, and it has a little more steep blue slope to it. Then when we go through and we take a look at the Henya-Greenstein scattering phase function parameters in our modeling, we can see the scattering difference. We can quantify that scattering difference. We see that the points for the 68501 and 10084 all tend to be up here in the positive C neighborhood, generally indicating strong backward scattering. Whereas for the, again, the very low maturity soil, the 71061, they all tend to be down here in this negative C neighborhood, which means that it's generally a forward scattering soil.
3 minutes.
Thank you. So what are the potential contributors? Well, again, much to our surprise, considering the results from a previous study, which I would direct you to read at some point in time on the simulants compared to the 10084, we did not see a strong correlation to the composition of the soil, the chemical composition of the soil. But we did see, of course, a difference between soils of one maturity and soils of a soil of a higher maturity. So we have coupled these measurements with scanning electron microscopy observations. And you can see a few of these here. These are actually scanning transmission electron microscopy, high angle angular dark field images of these soil grains. And you can see the space weathering differences. You can see that the 71061 does not have quite as much of the nanophase iron in its outer rims, whereas the 10084 and the 68501 very much do. And it's hypothesized here that those nanophase iron inclusions, along with some of the other space weathering properties, the agglutination of the more weathered soils, are contributing to making these a far more backscattering set of soils, whereas the 71061, not quite so much. There are other scanning electron microscopy observations that we've done with these soils that delve into the compositional effects, and I do have some of those images in backup slides. So in conclusion, the space weathering features, particularly nanophase iron, appear to have a strong effect, as near as we can tell, on the scattering direction for far ultraviolet light hitting these soils. But unlike our previous work, it does not appear at this time that there is a strong contribution due to chemical composition or overall mineralogy. Now, this does not mean that there may not be something along that line, because we still need to do more work with the scanning electron microscopy imagery to see if between the simulants that we measured previously and these soils, that there may be some kind of a dependence on composition going into the grain itself. So that might be something interesting to look into.
20 seconds.
And we plan to extend this work to hydrated lunar soil samples, where we cool these samples down the temperatures that are found in permanently shadowed regions at the poles of the moon and add water vapor. And I will put up this final slide with my contact info, and thank you very much.